And today we'll talk about the translation. What is the mechanism of translation in a detailed way? We know that this translation is simply the synthesis of proteins. Regarding the mechanism of translation, it is a multi-step process. Many steps are involved in the synthesis of polypeptide chain. First step is that is activation of amino acids. Activation of amino acids. As you know that translation means the synthesis of proteins. Though amino acids are the building blocks for the proteins, the first step for the translation is that is activation of amino acids. The second is charging of tRNA. Charging of tRNA. Third step is initiation of translation. Initiation of translation. Initiation of translation. Then we have elongation of polypeptide chain. Elongation of polypeptide chain. Then we have termination of translation and release of polypeptide chain. Termination of translation and release of polypeptide chain. Release of polypeptide chain and release of polypeptide chain. So these are the important steps which are involved in the translation process. Translation is a multi-step process. It involves several steps. First is activation of amino acids, because amino acids are the building blocks of the proteins. They need to be activated. Then we have got charging of tRNA. Then initiation of translation, elongation of polypeptide chain, and termination of translation and release of polypeptide chain. We will discuss these steps one by one. First, we will take this activation of amino acids. First step is activation of amino acids. Activation of amino acids. As we know that amino acids are the building blocks for the proteins. To get incorporated in a polypeptide chain, they need to be activated. How they are activated? They are activated by the reaction with the ATP. That means we have got amino acids plus ATP. This reaction takes place in the presence of enzyme amino acid tRNA synthetase enzyme. This is amino acyl tRNA synthetase enzyme. Amino acyl tRNA synthetase enzyme. This is amino acyl tRNA synthetase enzyme, and it will get activated amino acids. Activated amino acids. This is the first step, that is activation of amino acids. Amino acids they react with ATP adenosine triphosphate, and this reaction takes place in the presence of amino acyl tRNA synthetase enzyme, and what we get activated amino acids. Then these activated amino acids are being loaded on the tRNA. That is what is called as charging of tRNA. The second step is charging of tRNA. Charging of tRNA. This activated amino acids, you know that we have, this is the tRNA molecule. It is having the clover leaf model. This is tRNA molecule. This is the, so this is 3' hydroxyl end, and we have special nitrogen base sequence, that is CCA here, CCA at the 3' hydroxyl end of tRNA, and here we have got 5' end, and where we find phosphate, and also 7-methyl guanosine cap at the 5' phosphate end. We have 3' hydroxyl end, and this 3' hydroxyl end, it serves as an amino acid binding site. It serves as an amino acid binding site. It serves as an amino acid binding site. Then we have got this T-Cy-C loop. This T-Cy-C loop is for the binding of ribosome. It is called as ribosome attachment site. Then we have this is called as anticodon loop. Anticodon loop. This anticodon loop, it is having nitrogen base sequence complementary to the codons of the mRNA. It is called as DRM dihydrouridine RM. This is the binding site for the amino acid tRNA synthetase enzyme. So this is the tRNA molecule. The amino acid gets attached here. So this is the amino acid. This amino acid gets attached at the 3' hydroxyl end of the tRNA. This is T-Cy-C RM, where ribosome attachment takes place. This anticodon loop. This anticodon loop, and this is DRM, that means dihydrouridine OM, where the amino acid tRNA synthetase enzyme gets attached. So this is ribosome binding site. Ribosome binding site. This is anticodon, where anticodons are present. This is amino acid tRNA synthetase enzyme binding site, DRM, amino acyl tRNA synthetase enzyme binding site. This is amino acyl tRNA synthetase enzyme binding site. The typical structure of the tRNA molecule. This amino acyl tRNA synthetase enzyme, it binds the amino acid at the 3' hydroxyl end of tRNA by means of the covalent linkage. That means amino acid is attached to the 3' hydroxyl end of the tRNA by means of the covalent linkage by amino acid tRNA synthetase enzyme, and this is called as charging of tRNA. This is called as charging, or we can say that is charged tRNA. The tRNA having specific amino acid loaded on it is known as charged tRNA. And this charging of tRNA, that means the amino acid is being loaded on this tRNA by amino acid tRNA synthetase enzyme. And this amino acid tRNA synthetase enzyme, it is one specific for each amino acid. It is specific for each amino acid. Another important thing regarding it, that the relation between the amino acid tRNA synthetase and its tRNA, it is called a second genetic code. Second genetic code. What is second genetic code? The relationship of amino acid tRNA synthetase to its corresponding tRNA is called a second genetic code, and this second genetic code, it refers to the critical role of amino acid tRNA synthetase in maintaining the accuracy during the protein synthesis. This is what is called a second genetic code. So what is a second genetic code? Second genetic code refers to the relationship between the amino acid tRNA synthetase and its corresponding tRNA. And this relationship between the amino acid tRNA synthetase and its corresponding tRNA, what is known as second genetic code, it refers to the critical role in maintaining the accuracy during the protein synthesis. Then we have got, this was regarding the charging of tRNA. Then next step is, this is charged tRNA. Then now this charged tRNA having the amino acid loaded on it will be taken to the ribosomes where the actual protein synthesis takes place. So next step is, we have discussed activation of amino acid, charging of tRNA. Then third is initiation of translation. Initiation of translation. In initiation of translation, we have three initiation factors. We have three factors, what are called as initiation factors. Initiation factor first, initiation factor second, and initiation factor third. These initiation factors play an important role during the translation, initiation of translation. This initiation factor first, what is its role? It stabilizes the 30S ribosomal subunit. It stabilizes. We are here talking about the translation of the prokaryotes. It stabilizes 30S ribosomal subunit. 30S subunit of ribosome. Then initiation factor second. This initiation factor second, it binds to GTP. Binds to GTP and hydrolyzes. It binds to GTP and hydrolyzes GTP. Hydrolyzes GTP. This initiation factor second, it binds to GTP and hydrolyzes GTP. It also binds formyl methionine tRNA to the 30S mRNA complex. It binds formyl methionine tRNA to 30S mRNA complex. Then initiation factor third, this is responsible for binding of 30S ribosomal subunit to the mRNA. So it binds 30S ribosomal subunit to what? To mRNA. Under the influence of this factor, initiation factor, 30S ribosomal subunit binds to mRNA. And this initiation factor third is also responsible for preventing the premature binding of 30S and 50S ribosomal subunits. Prevents premature binding of prevents premature binding of. It was asked in a competitive exam, what is the role of initiation factor third? It prevents premature binding of 50S and 30S ribosomal subunits. So these are the initiation factors which were involved in the initiation of translation. So how this initiation takes place? This 30S ribosomal subunit binds to the 30S ribosomal subunit binds to the initiation factors, and this complex then binds to the ribosomes. So we have got initiation factors. We have got initiation factor third. It binds to what? It binds to 30S ribosomal subunit. That means small subunit of ribosome. 30S subunit of ribosome, and this complex then binds to what? It binds to mRNA. It binds to mRNA. So we have got five. This is 5' end of mRNA. This is 3' end of mRNA. We have codons on the mRNA. Obviously, it is AUG, it is initiation codon, and preceding to the initiation codon, there is a sequence which is called as Shine-Dalgarno sequence, AGG. This is Shine-Dalgarno sequence. This is Shine-Delgado sequence. It's called as Shine-Delgado sequence. This Shine-Delgado sequence was first described by Shine, two scientists, Shine and Delgado, and they said that it is this sequence of the messenger RNA which helps us in binding of mRNA to the ribosome. So this sequence is responsible for the binding of messenger RNA to the small subunit of ribosome. This Shine-Delgado sequence, it is a base sequence of 6 nucleotides. It is a base sequence of 6 nucleotides, 1, 2, 3, 4, 5, 6, and these are all purines, adenine and guanine, 6 nucleotides. They are preceding at the initiation codon present on the mRNA. This is in case of prokaryotes. The Shine-Delgado sequence in case of prokaryotes. Remember that the Shine-Delgado sequence, it is responsible for the efficient translation of the mRNA. Then we have got another, in case of eukaryotes, what we find? In case of eukaryotes, we have Kozak sequence. And this Kozak sequence was discovered by Kozak, in scientist whose name was Kozak, in 2002. And this sequence, it is also found around the initiation codon in the eukaryotic mRNA. And the sequence is GCC ACC AUG G. So this is what is called as Kozak sequence. This Kozak sequence is found around the initiation codon on the mRNA. And now both these sequences, whether it is Shine-Delgado sequence in case of prokaryotes and Kozak sequence in case of eukaryotes, they are responsible for the effective, accurate and efficient translation of the mRNA. And these sequences, whether it is Kozak sequence or Shine-Delgado sequence, Shine-Delgado sequence in case of prokaryotes and Kozak sequence in case of eukaryotes, it is responsible for the binding of messenger RNA to the ribosomal subunits, that is small ribosomal subunit. So we know that we have got initiation codons which are responsible for the initiation of translation, that is AUG and GUG. This AUG, it is an initiation codon, and same is the GUG, it is also an initiation codon. This AUG, it codes for methionine in case of eukaryotes. This AUG codes for methionine in case of eukaryotes, and it codes for formyl methionine. It codes for formyl methionine in prokaryotes. So AUG, it is an initiation codon. It codes for formyl methionine in prokaryotes, and it codes for methionine in eukaryotes. And this GUG, normally GUG codes for valine, but it codes for valine, but when it is present at the initiation position of the mRNA, it can code for the methionine. So this is an ambiguous codon. This GUG, it codes for valine when it is present at some other position on the mRNA, but when this GUG is present at the initiation position of the mRNA, then it codes for methionine. So it can also act as an initiation codon. Then we have got this then what happens, how initiation starts. We are talking about the initiation of the prokaryotes. Then formyl methionine, the formyl methionine tRNA, it will arrive at the P site of the ribosome, and the anticodon of the tRNA establishes linkage with the codon of mRNA at the A site, and the GTP is required for this linkage. So we can show it like this. we have, see this formyl methionine tRNA. Formyl methionine tRNA, it arrives at the P site of the ribosome. It arrives at the P site of the ribosome, and it is anticodon. Establishes linkage with the codon of mRNA. mRNA at the A site. And which initiation factor is required for this? This initiation factor second is required for the binding of formyl methionine tRNA to the ribosome, to the 30S mRNA complex. So we have like this, we can show it like this. We have for example, this is 5' 3' and this is messenger RNA, and we have codon AUG, initiation codon AUG. This is messenger RNA AUG. Obviously this is tRNA. It is having what? Formyl methionine. This is formyl methionine. This is tRNA. And tRNA is having anticodons. G will code for cytosine, and U will code for adenine, A will code for uracil. The anticodon of tRNA will establish linkage with the codon of mRNA, and this linkage is facilitated by the initiation factor second. So GTP is required for this. GTP required for this linkage. Now the larger subunit of the ribosome binds to the to this complex. So this is initiation of the translation. Now the larger subunit of the ribosome can bind to it. Remember that magnesium is required for the binding of larger subunit. This magnesium is required, magnesium ion is required for the binding of this. We have got transfer RNA. Then obviously this is small ribosomal subunit, small ribosomal subunit. I told you that this messenger RNA, it is having codons. They establish linkage with the anticodons of tRNA. This messenger RNA, it is bound to this small ribosomal subunit. This is bound to the small ribosomal subunit, which is facilitated by the initiation factor third. We know that this ribosome has three sites. One is called as P site, another is called as A site, and then we have E site. The first tRNA, that means this is first tRNA, first charged tRNA. What is here in case of prokaryote is formyl methionine tRNA. It arrives the ribosome at the P site of the P site, and then it establishes linkage with this anticodon, establish linkage with the codon of mRNA at the P site. Then the larger subunit can attach to the smaller subunit. Then this is a larger ribosomal subunit. It can attach to this initiation complex, and elongation process can proceed. And magnesium is required. This magnesium is required for the association of larger subunit of ribosome to the smaller subunit. This is smaller subunit of ribosome. So we have seen that this the first charged tRNA, it arrives at the P site of the ribosome, and this it is the anticodon of tRNA. It establishes linkage with the codon of mRNA at the P site, This is small subunit of ribosome, this is largest subunit. Then the largest subunit of ribosome will join to this initiation complex, and elongation process proceeds. This is what is called as initiation of translation. Then we have got another step, that is elongation of translation. Then we have got another word, elongation of translation. Elongation of translation. This elongation of translation, the ribosomes physically move along the mRNA in the 5' to 3' direction. So this is elongation of translation. Elongation of translation. We have 3 elongation factors which are involved in the this elongation of polypeptide chain. We can say that elongation of polypeptide chain. Elongation of polypeptide chain. First we have completed initiation of translation, then elongation of polypeptide chain. 3 elongation factors are involved in the elongation of polypeptide chain. Elongation factor, first elongation factor TU, first elongation factor TU, elongation factor TS, and elongation factor G. This elongation factor EF means elongation factor TU. U means unstable at high temperature. Same is the case, we have elongation factor TS. S means stable and T means temperature. That means this factor is stable at high temperature. And elongation factor G. G refers to the G protein. It is a GTPase. So this elongation factor TU, it binds to the GTP and hydrolyzes GTP. It binds to GTP and hydrolyzes GTP. Hydrolyzes it. This elongation factor TU, it is responsible for binding of amino acid tRNA at the A site of the ribosome. It binds amino acyl tRNA at A site of ribosome. A site of ribosome. Then this elongation factor TS, it activates or it stimulates what? It stimulates elongation factor TU, because elongation factor TU is important. It hydrolyzes GTP, it binds to GTP and hydrolyzes GTP. It is also responsible for binding of amino acid tRNA at the A site of the ribosome. This elongation factor G, it has role in translocation. It is also GTP dependent protein. It is GTP dependent protein. It is also GTP dependent protein. Now how this elongation process takes place, we know that this is mRNA and this is ribosome. The ribosome, it physically moves along this is the 3 sites of the ribosomes. We have A site, this is P site, and this is E site. These are the 3 sites. There are 3 sites in the ribosome: A site, P site, and E site. A means amino acid acceptor site, P site is peptidyl site, and E site is exit site. So only the first amino acid, first charged tRNA arrives at the P site of the ribosome, whereas all the other tRNAs, that means second, third tRNA, fourth charged tRNA, they arrive the ribosome at the A site. That is why A site is called as amino acid acceptor site or amino acid acceptor site. P is the peptidyl site and E is the exit site. So first tRNA, first charged tRNA, that is formyl methionine tRNA in case of prokaryotes and methionine tRNA in case of eukaryotes, they arrive the ribosome at the P site, whereas rest of the tRNAs, they arrive the ribosome at the A site. So this mRNA, it physically moves along the this is ribosome. This is mRNA. This ribosome physically moves along the mRNA in the 5' to 3' direction. That means this ribosome, it moves on the mRNA in this direction. That means in the 5' to 3' direction. As a result of it, when it moves in the 5' to 3' direction, all the codons which are present on the mRNA, they will get exposed at the A site, and the polypeptide chain grows. So we can say that this second tRNA will arrive. Second tRNA, or we can say that second amino acid tRNA, charged tRNA, will arrive at A site of the ribosome. Will arrive at A site of the ribosome. It would be like this. We have this messenger RNA 5' to 3'. This is messenger RNA. Now we have these ribosomes. This is P site of ribosome. This is E site of ribosome, and this is A site of ribosome. As we know that first tRNA, it is present at the P site. This is formylmethionine, that is amino acid first. It is present at the P site. Now the second charged tRNA will arrive at the A site. So this is second charged tRNA. It will arrive at the A site. So it would be like this. Amino acid second. Now what will happen? There will be a translocation. See what will happen? This the covalent linkage between the first amino acid and tRNA is broken, and there is an enzyme that is called as peptide transferase that forms a peptide bond between the first amino acid and the second amino acid at the A site. So two process. Then this tRNA at the A site is translocated to the P site. This is what is called as translocation. So what will happen? This covalent linkage between the tRNA and its amino acid is broken, and then peptide transferase, then this peptide transferase, it forms the peptide bond between the first amino acid and the second amino acid at the A site. It would be like this. Then we have got 5', this is 3'. This is small ribosomal subunit, large ribosomal subunit. We have P, A site, E site. Then the linkage between the first amino acid and its corresponding tRNA is broken. As a result of that, it is broken, and a peptide transferase enzyme, it establishes a linkage between the first amino acid and the second amino acid. That means this amino acid will get transferred to this tRNA which is present at the A site. It would be amino acid first, amino acid second. So now the tRNA at the A site, it is having two amino acids, and these two amino acids are joined by what? Peptide bond. This peptide bond is formed by enzyme peptide transferase. This peptide transferase is an important enzyme. It is a ribozyme. That means it is an RNA enzyme. It is the RNA enzyme that is ribosomal subunit, or RNA of large ribosomal subunit will act as a ribosomal RNA of large ribosomal subunit, or RNA of large ribosomal subunit acts as a ribozyme. So peptide transferase, it is a ribozyme, it is an RNA enzyme. And then what happens? Then this we have this empty tRNA at the P site. Now this tRNA at the A site is translocated to the P site, and the tRNA which is unloaded at the A site, at the P site, it is being shifted to the E site. This is known as translocation. Translocation, what do you mean by translocation? It simply means movement of charged tRNA from A site to P site, coupled with the movement of codons. And when there is the movement of tRNA, in addition to this, it is being coupled with the movement of codons of mRNA, so that all the codons will get exposed at the A site. So in addition to this, there is coupled with movement of codons of mRNA. This is what is called as translocation. And this translocation, it requires enzyme translocase and GTP. So elongation factor S, it also helps in the translocation. And then in the next step, what happens? This is, we'll show it here. This larger subunit of ribosome, this is small subunit of ribosome, this 5' 3' messenger RNA. We have P site, we have A site. Now this tRNA at the site, it is being shifted to the P site. It will be like this. So this is amino acid second, amino acid first. Obviously, this is uncharged tRNA at the E site. It will slip away from the ribosome. Then the another tRNA will arrive. This is a third tRNA. It will arrive the ribosome at the A site. For example, it is amino acid third. It will arrive the ribosome at the, this is amino acid third. This amino acid third, then third amino acid arrives the ribosome at the A site. Then what happens? Again, the covalent linkage between the tRNA and its corresponding amino acid is broken, and then this dipeptide shifts to the tRNA which is present at the A site, and now there would be 3 amino acids on the tRNA which is present at the A site. And this is the process of arrival of charged tRNA, translocation. It is being repeated, and the polypeptide chain grows. It would be like this. Again, it would be like this. This is small subunit of ribosome, large ribosomal subunit. This is mRNA, P site, E site, P site, A site. This is E site. This at the P site, the linkage between this dipeptide and tRNA at the P site is broken. So it would go here. So we have this configuration. tRNA at the amino acid, tRNA at the site A, it is having amino acid third. Now this will be having 3 amino acids. This is amino acid second, this is amino acid first. So again this tRNA, it will get shifted to the P site, and the tRNA at the P site will get shifted to the E site and will slip away. In this way the translation process grows. So we can say that the arrival of charged tRNA, then arrival of charged tRNA, peptide bond formation and translocation, this process repeated and polypeptide chain grows. We can say that three processes are important in the elongation of the polypeptide chain. One is arrival of charged tRNA plus peptide bond formation, peptide bond formation, peptide bond formation, peptide bond formation plus translocation. Translocation, they are repeated and polypeptide chain grows. These are repeated and polypeptide chain grows. And polypeptide chain grows. These three processes, arrival of charged tRNA, peptide bond formation, translocation, they are repeated and polypeptide chain grows. Remember that for the incorporation of each amino acid, 1 ATP and 2 GTP molecules are required. 1 ATP and 2 GTP molecules are required for the incorporation of one amino acid in a polypeptide chain. Then we have got 1 ATP and 2 GTP molecules are required for the incorporation of each amino acid in a polypeptide chain. Then we have got last one, that is termination and release of polypeptide chain. So we have got last one, that is termination and release of polypeptide chain. So arrival of charged tRNA, peptide bond formation and translocation, they are repeated and polypeptide chain grows. Then last one we have got termination of translation and release of polypeptide chain. Release of polypeptide chain. Remember that three termination codons. We have UAA, UAG and UGA. These are known as termination codons. They do not code for any amino acid and the polypeptide chain stops. This UAA is also called as Ocre codon. It's called as Ocre amber codon, and UGA is called as opal codon. This is the technically this UAA is called as Ocre codon, UAG amber codon, UGA opal codon. These are called as stop codons or termination codons, or they were earlier called as nonsense codons. They do not code for any amino acid. That when they arrive at the A site, of the ribosome, they signal the stop, your halt, your termination of the translation, and the growth of the polypeptide chain stops when these termination codons arrive at the A site of the ribosome. In addition to this, there is a dissociation factor and decrease in magnesium concentration. This dissociation factor and decrease in magnesium concentration is responsible for the dissociation of the ribosomal subunits. That means ribosomal subunits, they dissociate under the influence of the dissociation factor. It's a protein, and decrease in the magnesium concentration. Magnesium is required for the assembly of the ribosomal subunits, but when this magnesium Mg2 plus concentration will decrease, these ribosomal subunits will get disassembled. And then there are important release factors: release factor first, release factor second, and release factor third. This release factor, first and release factor, release the polypeptide chain from the catalyze the release of polypeptide chain from terminal tRNA. These protein is release factor first, release factor second, and release factor third are responsible for the release of polypeptide chain from the terminal RNA, while as release factor third, it stimulates release factor, first and release factor second. In eukaryotes we have single release factor, eukaryotic release factor. This in eukaryotes there is a single release factor that is called as eukaryotic release factor. Now we know that this these release factors, they bind to the termination codons, they recognize the termination codons, bind to the termination codons, and induce the peptide transferase to release the polypeptide chain from the terminal tRNA. This is the function of the release factors. And then this polypeptide chain, it moves out of the ribosome through a groove in a large ribosomal subunit. So we have got this, large ribosomal subunit, this is small ribosomal subunit. There is a groove in the large ribosomal subunit. It's a groove, tunnel in the large ribosomal subunit, and this polypeptide chain, it emerges from the groove. This is the growing polypeptide chain, nascent polypeptide chain, we can say, nascent polypeptide chain. Or nascent polypeptide chain. The newly synthesized polypeptide chain emerged through a groove or tunnel in a large ribosomal subunit. This is large ribosomal subunit. So in this way, this ends the termination of the translation. So remember that translation, it takes place in steps. We have activation of amino acids, charging of tRNA, then initiation of translation, elongation of polypeptide chain, and termination of translation. So these are the important steps which were involved in the translation mechanism. Hope you have understood. Thanks.